AMMF's involvement with the research team in Edinburgh goes back over a number of years. And this afternoon, we'll hear from some truly dedicated researchers who are doing what they do to help us understand cholangiocarcinoma better. And through that understanding, to find ways to an improved future for all diagnosed with this disease. Now, before we get going, there's just a few housekeeping points, and these are all on the tips sheet you will have received with your link. The event will be recorded. We've just heard that begin, but the recording will stop before the Q&A session. During the tour, please keep your microphone muted and your camera off, but do turn it back on again for the Q&A, because it really is lovely to see you all. Questions can be via the chat box or raise your hand. That's the little yellow Zoom one, if you'd like to ask your question verbally. Now I'm going to hand you over to Robin, who will introduce you to this afternoon's stars and who will guide us through the tour. I wish everyone a very enjoyable and informative time. And until we meet again for the Q&A session, over to you, Robin.
Thank you very much, Helen, and welcome everyone. My name is Robin Morton. I'm the Communications Manager at the Centre for Regenerative Medicine at the University of Edinburgh. So on behalf of our director and the director of the Institute of Genetics and Cancer, welcome to our tour. What I'd like to say is my boss is the director of our centre is Professor Stuart Forbes. And in fact, two of the presenters today started their research careers with Stuart. And in fact, when we were chatting earlier, credited Stuart with getting them into this field of research. So it's really nice for me actually to have that conversation with them, because I've heard their names so often in my job. My job is really to tell people about the research that goes on in our centre. But I've got three other colleagues with me on this call. Comms people never go in ones, we always go in fours. So we've got Steph Sinclair, we've got Dee Davison and Connor Warnock. So hello, give us a wave, Steph. So Connor's going to be working in the background. So if I'm saying, Connor, please start a video, then you know who he is. I don't know if we can see Connor just now, he's not on my screen anyway. I'd also like to introduce you to our researchers. So on the call just now, we have Luke Boulter, Annabelle Martinez-Lyons, and Rachel Guest. And virtually, we also have Ben Dwyer, who's now based in Perth, Australia. So I'm going to do a couple of things, one of which is I'm going to share my screen. And if I try and talk at the same time as do this, I always get it wrong. So I'm going to start by doing that. And then I'm also going to make sure I can see everyone who's got their video on. It's a little bit more reassuring when I can actually see people instead of just a blank screen. And if I look off to the side, it's because I've got people on my left and I've got my virtual tour in front of me. So please excuse that. So as I say, my job is to communicate the research that goes on in our institute, and Dee and Steph both do that job as well, but in slightly different areas. So if you were coming to our centre, and we very much hope that you'll be able to in future, we would be welcoming you in our reception area, which you can hopefully now see ahead of you. Can everyone give me a thumbs up if you can see that? Excellent. Always good to check. So let's head into the centre. This is me talking to one of our PhD students at the time, now a postdoc in the centre, Sophie. Just to highlight Sophie, because she's in every picture, but the reason she's in every picture is that if you want to go on this tour yourself, or if you want to share this tour with others, then Sophie would be your virtual guide by clicking on those white triangles in the picture. So let's head upstairs. This is, I should say, welcome to the University of Edinburgh. Really, we're showing you just one of our centres, and the College of Medicine and Vet Medicine is based right across the city of Edinburgh. So D, for example, if we weren't sitting in our homes, we'd be sitting in our centres at opposite sides of the university, but we very much collaborate and work together right across the university. So let's head upstairs. So first of all, just heading into our seminar room, here's a typical lab meeting. Luke may remember having lab meetings with Stuart in our CRM seminar room. And I think that's a good point to introduce Luke, and Connor is going to share a little video of his research just now.
Hello, I'm Luke Bolter and I'm a principal investigator at the MRC Human Genetics Unit in Edinburgh. We're part of Edinburgh University and part of the Institute for Genetics and Cancer, which involves the Human Genetics Unit where I am, but also a cancer centre and a centre for disease mechanisms as well. My lab works on a number of things, but we're really interested in the bile duct. The bile duct is this tube that runs through the liver, or set of tubes that run through the liver. And we're really interested in how those tubes form in the embryo, so as you develop as a fetus, but also how those tubes then regrow during disease, and importantly, how those regrowing tubes can become cancers. We think by understanding the processes of how the bile duct grows and changes over lifetime, that we can understand some of the mechanisms that are in place when a cancer forms. And by understanding those mechanisms and those processes, we hope to develop new drugs for treatment of cholangiocarcinoma. Cholangiocarcinoma is a really challenging cancer to work in for a number of reasons. A lot of patients come to the clinic with very established disease, and so it's very hard for us to find ways to intervene early. Cholangiocarcinoma as well is very diverse, so the tumours come from different parts of the liver, they look different, they have a different set of changes in their DNA, and so all of those things together make cholangiocarcinoma a very difficult tumour to treat. Currently, most patients will receive chemotherapy if they're not eligible for surgery, and because of that, the survival for cholangiocarcinoma following a diagnosis is quite poor. What we hope to achieve is an improvement in that with our new methods of developing drugs and medicines for the treatment of cholangiocarcinoma. My lab has been working over the last few years to really develop our tools and our systems that we can use to understand how cholangiocarcinomas grow. Particularly, we want to know what causes the cancer to start. So if you do have underlying disease, or even if you don't, why has a tumour begun to grow in your liver? We use a series of animal models for this, so genetic tools in mice and rats, but also human samples as well, so donated very kindly by patients, which we can grow in the lab and use to test new therapies and new medicines, with the expectation, of course, that ultimately we'll be able to put those back into the clinic. It's a very difficult question to ask yourself what you brought to the field. I think ultimately we have developed a series of tools that other researchers can use. So we've tried to really open up cholangiocarcinoma research to not just our group, but other groups from Edinburgh and elsewhere in the UK and the world, actually. We also have been quite successful in developing some new medicines. So medicines that we developed in 2015 have now been put into a clinical trial, and hopefully those will show some efficacy in patients, and we'll be able to use those to treat a broader spectrum of patients who perhaps aren't eligible for current treatments.
So let's continue the tour. I can't see it. So show video panel. There we go. Let's continue the tour. Hopefully you can see it now again. Yeah. So let's head over maybe Luke and Rachel will recognise the yellow area, and maybe some former colleagues. Now, at this point, just to introduce, this is where Stuart's group sits in the write-up area, and then Stuart's laboratories are through the window. And as I mentioned, Dr. Ben Dwyer was very much part of Stuart's group until I think the end of 2020, at which point, as often happens with Stuart's group, he headed off to start his own group in Australia. So he's been very successful in his research and is now very active researching in Australia. But before Ben left, we managed to catch an interview in which he describes some of the research that he did, supported by AMMF. Connor, could you bring up Ben's video?
Hello, my name is Robin Morton from the Institute for Regeneration and Repair at the University of Edinburgh. Today I'm joined by Dr. Benjamin Dwyer of the Forbes Group in the Centre for Regenerative Medicine, and he's going to be telling us a little bit about his recent paper in the Journal of Hepatology. Ben, in brief, could you tell us what this paper is about?
So in this paper, we define a mechanism by which cancer cells interact with their environment. We looked at a large number of clinical samples to show this is clinically relevant. We looked at mouse and rat models to manipulate this pathway and found that by inhibiting this pathway, we can prevent tumor growth.
So why is this work important?
This work is important because cholangiocarcinoma is a cancer that's extremely resistant to treatment and hard to detect early. So new treatments are urgently required. We show that the pathway we've discovered is activated early in tumor development and can be targeted to limit tumor growth. So we think this might be a potential new target for therapy for cholangiocarcinoma.
Could you describe cholangiocarcinoma and the environment around the tumor that's so important to this work?
Yeah. Cholangiocarcinoma is a tumor that resembles the liver bile duct epithelium. It can arise from various parts of the bile duct, inside or outside the liver. It's incredibly resistant to therapy and often diagnosed at late stage, so there's extremely poor survival. And this is mediated by the interaction between the cancer cells and the tumor environment. So the tumor environment is composed of cells like macrophages and cancer-associated fibroblasts and provides signals that promote tumor growth and resistance to chemotherapy. So given the resistance of this tumor to therapies, many labs are looking to target the environment of the tumor to improve therapy responses.
So how is AMMF, the cholangiocarcinoma charity, involved in this work?
So AMMF, the cholangiocarcinoma charity, have been excellent in providing funding support for this project, and they also do a lot of work advocating for cholangiocarcinoma patients. Without the help of AMMF, this work wouldn't have been done. Also had the opportunity to speak at their conference and interact with cholangiocarcinoma patients and their families, which is hugely important to me. We don't do this work just because of the interesting science. We do it to make a difference to patients. And AMMF is excellent at bringing patients together with researchers and clinicians.
Thank you very much, Ben, for joining us to talk about your recent paper, and very best of luck with this work in the future.
Great. Thanks very much, Robin.
We'll continue the tour. So in a lot of our research, cell culture is very, very important. So a lot of research involves growing of cells and keeping them alive. So this is just to take you into our cell culture room, which is very typical of cell culture rooms around the university, both in CRM and Institute for Genetics and Cancer. And I think at this point, I'm going to introduce a little video by Molly, who unfortunately couldn't be here today, but has sent this video. So, Connor.
I'm Molly, and I'm a final year PhD student in Luke Boulter's lab, and I've been fortunate enough to have my entire PhD project funded by the AMMF. So I work on intrahepatic cholangiocarcinoma, and this is the cancer of the biliary tree that AMMF are focused on. So more specifically during my project, I've been looking at the changes in DNA that occur in this cancer with the idea that if we can use animal models to look at how these genetic mutations affect the animals, then this might be applicable to the human cancer too. AMMF have allowed me to carry out all of my scientific research, and they've actually allowed me to publish a paper in a very highly respected journal, which is really exciting. I'm really grateful to them for providing this funding.
There we go. So we've moved to a facility called FACS. So this is fluorescent activated cell sorting. And in FACS, there are these pieces of equipment, something between 250,000 and 500,000 pounds worth, that can essentially sort different cell types. And once you have these different cell types, you can then do whatever experiments need done. Also very important to much of the research that we do is microscopy. So this is me just trying to find my way through this tour to find the microscope room. There we go. So in confocal microscopy, again, very expensive pieces of kit that give us these incredible high definition images of these cells and tissues that are so important to our research. So again, now someone we do have on the call is Annabelle, and Annabelle's going to tell you a little bit about her research using this kind of equipment.
Hi, my name's Annabelle. I'm a postdoc in Luke Bolter's group. My research focuses on the early stages of cholangiocarcinoma formation and progression. To study cholangiocarcinoma, we use mouse models of cancer. In these, we trigger cholangiocarcinoma formation with genetic changes as well as liver damage. We use high-resolution microscopes to visualize changes in bile ducts, from healthy ducts, like these, to cancerous ducts, as seen here. In this tumour, we are looking at a bile duct marker in white, cancerous cells in red, and finally dividing or proliferating cells in cyan blue. We use imaging to look at how cancers form and how they grow over time. This information helps scientists to develop therapies that target specific parts of the cancer growth process. This research relies heavily on our access to specialised microscopes and imaging tools, which are available as part of the Advanced Imaging Resource Facility here at the Institute of Genetics and Cancer.
As I say, at a later date, you can come back and take this tour for yourself. What I'm going to do is just switch scenes and take you up to our breakout space. The wonderful thing about the Centre for Gender Medicine is it's very, very much a light and airy atmosphere and lots of glass, and that's really to encourage collaboration. So collaboration is just vital to what we do. And if we just pop back to our seminar room, you can see in the distance there we have Edinburgh's main teaching hospital, and so the Edinburgh Royal Infirmary, or Royal Infirmary of Edinburgh. And in there, for example, Stuart Forbes has his liver clinics. We have clinical research facilities. So really, we're perfectly placed to translate our research into the clinic. And again, we're lucky enough to have Rachel Guest on the call today, and Connor is just going to show this final video of Rachel talking about her work.
My name's Rachel Guest. I'm a hepatobiliary surgeon at the Royal Infirmary in Edinburgh, and I'm also a clinician scientist based here at the Edinburgh Cancer Research Centre, funded by the Wellcome Trust to research bile duct cancer or cholangiocarcinoma. As a clinician, it's been hugely frustrating treating patients in the past where we've had very few treatments available to offer patients. And we now appear to be moving into a hugely exciting time in bile duct cancer research, where we're discovering new targetable mutations in bile duct cancer, which have therapeutic options to treat patients. My work in the lab focuses on one particular mutation, which we're hoping offers a therapeutic option for patients. We're hugely grateful to the AMMF charity for supporting our research into bile duct cancer, and we're gaining momentum as a liver cancer hub here in Edinburgh, where our research can directly impact and help improve the clinical outcome for these patients.
So just back in reception. So thank you very much for joining us on this virtual tour. And I hope what we've shown you is that collaboration is absolutely key. And that collaboration doesn't just extend between scientists and clinicians, it also extends to organizations like AMMF. And we're incredibly grateful for their support and for their collaboration. So on that note, I'd like to pass back to Helen.
Thank you very much, Robin. And thank you, a heartfelt thank you to everyone involved for putting on this tour for us and for taking the time out of all your busy schedules to organize this and to do the recording of those really great presentations. I think hearing about the impressive research that's being carried out here reminds us of how much we still need to learn about cholangiocarcinoma, and also just how necessarily painstaking and time-consuming research is. But bit by bit, we are making progress. We are seeing things that 10, 15 years ago, we possibly wouldn't have even thought of. It's really been invaluable for us to have this glimpse behind the scenes and to get just a taste of what's being done. And also to see and hear from some of the wonderfully dedicated team you have in Edinburgh. I mean, what we do as a charity in raising awareness and what our wonderful fundraisers do in supporting us financially means that we can encourage researchers to do what they do best, and that is to help us to find the answers that will one day lead to a better understanding and eventually to a cure for cholangiocarcinoma, because that is the ultimate goal. So thank you once again. It's been fantastic to see this, and actually, you know, really encouraging that the help that AMMF as a small charity, the help that we can give you, and which has been in many times sort of almost pump priming grant funding, but to see what it's leading to is fantastic. So thank you everybody for what you're doing.